Hey y'all, and welcome to Tech Fact Friday. Today, we're finishing up our 3-part series, digging into the history of biotechnology. Last time we left off around the beginning of the 1970s, and if you thought in the previous episode we had a lot of biotech discoveries in a short period of time, just wait until you hear this. The 1970s was a time for enormous, revolutionary growth in the field of biotech. It would take a whole episode just to list out everything between the 70s to now. But picking back up in the mid-1970s, we get the chickenpox vaccine and the creation of synthetic monoclonal antibodies, both products vital improvements to immunity. Monoclonal antibodies have become a recent addition to our daily lexicon, but what do they do? Unlike a vaccine, which is typically given before an infection, monoclonal antibodies are given during an active infection or disease process. It essentially boosts the immune system and helps fight the specific disease the antibody was created to attack. But it can also be used for detection, like at-home pregnancy tests, or to treat things that aren't infectious, like migraines and high cholesterol. Shortly after this, in 1978, we got synthetic human insulin, which was approved for production by the FDA in 1982. Remember where we left off on part 2 with recombinant DNA? This insulin is the result of that technology. And while we're recalling information, in part 1 we said biotech wasn't just about genetics. It encompasses a lot. Well, in 1980, a microorganism was developed to help the environment when man-made catastrophes happened, specifically oil spills. This organism's sole purpose was to eat oil. So here we see a coalescing of environmental and genetic sciences on this end of the biotech field. Continuing on through the 80s, we got genetic fingerprinting, the identification of the AIDS virus, and plants genetically engineered to resist bugs and disease. 1990 saw the launch of the Human Genome Project that sought to identify and map out the entire human genetic code, basically the programming code for a person. Genetic-based diseases like certain cancers or Alzheimer's were able to be identified, and therefore testing for treatment for these diseases could begin. This project was completed in 2003. We're only in 1990, and we have another good 30 years to cover before we can start talking about the future. So in order to have time to talk about that, we're going to speedrun the next 30 years and hit the highlights. Here we go. Hep C testing for blood to ensure safe transfusions, laboratory-created human skin, synthetic DNA, cloning of a sheep, genome sequencing of microorganisms and animals, the ability to produce embryonic stem cells from skin cells, the first completely artificial DNA molecule, further identification of genetics in Alzheimer's, synthetic genome duplication independent of human interference, the development of a full trachea from stem cells, which was then transplanted into an actual human, 3D printed heart on a chip that mimics heart tissue, gene modification that allowed tomatoes to yield more, and brain-to-machine communication. Whew, okay. With all of those mind-blowing breakthroughs, what could possibly be on the horizon of biotechnology? According to the Department of National Intelligence, we could see the eradication of the most common diseases, since scientists are increasingly treating genetic instructions as a form of computational code. This could lead to treatments and therapies bioengineered specifically for an individual. Additionally, artificial intelligence is starting to play a larger role in biotechnology, specifically with identification of disease. For instance, it could be used to sort quickly through patients' data to identify groups of people that a particular treatment would be the most effective for. AI and machine learning could also be used to help scientists make decisions on what drug experiments to perform, which would lead to faster drug development since time wouldn't be wasted on non-viable experiments. Different areas of biotechnology that are likely to see new developments soon are environmental sciences and food. Genetic modification is already being used for some food products, but as climate change affects plant production, we'll likely see the results of efforts to create plants that are more heat resistant, retain water better, and can produce rapidly in a short grow season. Along a similar line, we'll probably also see a trend towards more waste-eating organisms, like the oil eaters mentioned earlier in the video. The organisms will likely be geared towards wastes like plastic and styrofoam, that can't break down naturally. In fact, plastic-eating organisms have already snagged their first job with a plant in France that breaks down PET-based plastic. Based on this information, it seems like the biotechnology of the future will be geared towards continuing to eradicate disease, including rare genetic diseases, giving longevity to human life with a higher quality of that life than in generations past, and making our food supplies more tolerant to climate change. That's all we have time for today. Thanks for joining us for this deep dive into the history of biotechnology. If this is your first time joining us, go back and watch the first 2 episodes in this 3-parter. Also, while researching this topic, we found a video of the 3D printed heart on a chip that we think you'll enjoy. The link for that is in the description. Check it out, it's really cool. Remember to share, like, and follow for more content like this. Until next time, it's time for an upgrade, a technical upgrade.